Hello, and welcome to the thematic session, Advancement and Innovation in Eye and Vision Research. This session is contributed by the Centre for Eye and Vision Research, or CEVR, an InnoHK-funded initiative that brings together researchers from the Hong Kong Polytechnic University and the University of Waterloo in Canada. My name is Ben Thompson, and I'm the CEO and Scientific Director of CEVR. Vision impairment affects over 2 billion people worldwide and is a major global health concern. Today, we will highlight research breakthroughs that will help to reduce vision loss in Hong Kong and worldwide. We will focus on two leading causes of visual impairment. The first is myopia, or short-sightedness, which is projected to affect over half of the world's population by 2050. The second is retinal degeneration, which can cause profound vision loss and represents a looming health crisis as the world's population ages. Today's speakers are globally recognized scholars who are all researchers within the Centre for Eye and Vision Research. The session is split into two parts, and part one is on the topic of myopia, from research and innovation to therapeutics delivery. And I'm very happy to introduce our panel speakers. From the School of Optometry at the Hong Kong Polytechnic University, Professor He Ming Guang and Professor Carly Lam, and from the School of Optometry and Vision Science at the University of Waterloo, Professor Lyndon Jones. So now I'd like to invite Professor He to share on myopia control, low energy red laser treatment.
Yeah, thank you so much for your kind introduction, Ben. My name is Ming. I'm just newly joined the faculty in the School of Optometry and Polytechnic University. I'm also a co-principal investigator at SIVA in Hong Kong. It's a great pleasure again to join this Asia Summit of Global Health. Today, I'm going to share with you we call it low energy red laser treatment as a new alternative to control myopia. Can I have my next slide, please? Yeah, before everything, I should declare a conflict of interest. We hold the granted patent as a method for increasing blood flow and metabolic rate on the ocular fundus. And then I'm also a director and shareholder of the company, or the manufacturer for the technology I'm going to mention. Can I have the next slide? Myopia is a huge problem. So for every optometrist or any pediatric ophthalmologist, we all understand the idea of myopia is really excessive axial elongation. So people develop myopia from memetropia. So this and then for those already established myopia, the purpose is really to control the progression so that the children will not become high myopia when they're turning to 18 years old, say adulthood. Because the purpose of this myopia control is to minimize the risk of developing, we call it MMD, myopic macular degeneration, for example, diffuse atrophy and macular hemorrhage in the future. So myopia control is about prevention of onset and also control the progression. Can I have the next slide, please? A couple of years ago, we run a clinical trial in China. So in this 3-year clinical trial, we proved that increase time spent outdoor, like say every day, you spend additionally 40 minutes per day, will reduce or avoid the onset of myopia by at least 20%. So can I have the next slide? So the idea is how to extend further so that the children can expose to longer outdoor time. So from 40 minutes additional every day to hopefully 2 hours. This is really a challenging task. So in China, we actually established this, we call it sunlight classroom, as a way to further extend the duration of outdoor exposure. While sunlight is free of charge, but then the challenge is how to maintain the temperature in a sunlight classroom is very expensive and also is pragmatically challenging in most of the school setting. So the question is, can we develop something that can directly or locally emit the light on the retina as a way to treat or control myopia? So this is called local retina therapy. So is this a new treatment option? Can I have the next slide, please? So then the technology is really is a home use device, as you can see in the picture. It's a device that can emit red lights, 650 nanometer, and by a laser semiconductor. So the device actually at the bottom is a small computer. So with this small computer, you can access internet with the assigned username and password. And then you log on the system online, and then at the back end, we will double check the username and password, and then you allow you to turn on the lights. So you are allowed to turn on the lights only when you have a username and password that match the database in the back end. So with that, the patients at home, they can turn on the lights 2 sections in a day and 3 minutes per section. So we recommend the children have this treatment at home 5 days a week. So this is the laser, we call it laser diode generated red light. Is a class 1 laser devices, and is safe and no possibly eye damages. Can I have the next slide? So then we run a clinical trial to prove the efficacy. So as you can see from this figure, so if in a control arm is a single vision spectacle, so that in 1 year time, one year clinical trial, so in 1 year time, the control arm got 0.4 millimeter axial elongation. But then in the intervention arm, the red light arm, the axial elongation is only 0.1, so representing around 70% efficacy in controlling axial elongation. Can I have next slide? And then the secondary outcome is a spherical equivalent progression in diopters. So in the control arm, we have a 0.8 diopters progression in the control arm, and then in the intervention arm is only 0.2 diopters progression in a year. So this is about 80% efficacy in controlling myopia. Can I have the next slide? Even more interestingly, because the system actually allows us on the back end, we can observe what's the compliance of the specific study participant. So when the compliance on the study participant improved from 50% or reaching 75% compliance, so the efficacy on the axial elongation can be 80%, and then in the spherical equivalent progression is nearly 90%, 90% control on the spherical equivalent progression. Can we next slide? So we did not observe severe adverse events, but then when we initiated the study, there 2 children felt the light is too bright at the right beginning, and then we just decided allowed them to withdraw from the study. But once the child or the children initiated treatment, so in 1 year time, no one really complained this light, red light is too bright, and then withdraw from the study. And then there's about 3 children lack of cooperation, and 1 committed to ortho-K in the treatment arm. Can I have the next slide? So then we also have an opportunity to call this patient come back at 2 years' time. So initially, the study is a 1 year clinical trial, but then in 24 months or 2 years, we ask the patient come back. So there's a different scenario. So we can see that if for the children who continue the treatment on the 2nd year, we can see sustained myopia control, as you can see on the bottom line, or the red line. And then, but then once the children stop treatment, I mean, in the control, in the intervention arm, there's some moderate rebound effect. So and then in the control arm, once the child or children initiate red light, it threatens the axial elongation. And then in the spherical equivalent, we can observe very similar rebound, sustained effect. Can I have the next slide? And something interesting is that consistently in this study cohort, we observed significant axial shortening. We're talking about 0.05 millimeter shortening, and it's as common as 1 out of 5, like 20% axial shortening in the 1 year visit. Can I next slide? So this is a typical interesting case. It's actually a pair of monozygotic twins, female. And then on the left-hand side is a twin in the intervention arm. We observed 0.2 millimeter axial shortening, and then we observed significant choroidal thickening. And then on the right-hand side is a twin side in the control arm. We observed this consistent myopia progression. Can I have the next slide? Next slide. We try to prove that safety. So this we recently performed we call it light safety assessment, and then in the FDA accredited clinic lab, and then we proved this is group 1, very safe to use, and then the device has also obtained regulatory approval from CE mark and so on. So in summary, there's some ongoing international clinical trial in Japan, US, and also Australia. Next. There's but the possible mechanism may be just relevant to sacral hypoxia. And then the next slide. In conclusion, this home use device is a repeated low-level red light treatment, very simple to use, and then you could create a very stable and reliable myopia control effect, and with we didn't observe documentable functional and structural damages. Thank you so much.
Thank you very much, Professor. That's extremely exciting results from this technology. I wonder, you mentioned some ongoing trials. So how many people are using this device at the moment? And you mentioned a few side effects in your presentation, but have you detected any other side effects from the use of this treatment?
So at the moment, there's in China alone that every day we have around 70 to 80,000 children using it. So we have our system, the company have a system to monitor all these severe side effects. And then if anyone got a compromised best corrected visual acuity, they got to report it. And then or someone we got to extend it after image, they will also report it. So at the moment, we observe a handful of light injury, and then we are submitting and we'll be publishing papers reporting one light injury. We call it light injury cases, and then but once we stop the treatment for 3 months, and then the damage is recovered fully.
Wow, so tens of thousands of people already using the technology. You mentioned that a lot of the use at the moment is in China. Do you have any reason to think that there may be any ethnic differences in the response to the therapy if the treatment is used elsewhere, for example?
It is a great question. So actually in both in Australia and UCSF in US, we're running clinical trial to prove the efficacy in multiple ethnic groups. Actually, very interesting. We observe a much better treatment effect in the Caucasian population.
Oh.
So yeah, but then the India population, we observe very similar treatment effect.
Very interesting. Professor, thank you very much for sharing this new technology.
You're welcome. Thank you so much.
So staying on the topic of myopia control, I'd now like to pass to Professor Carly Lam, who will speak on myopia control, a journey from disc to DIMS.
Thank you for the opportunity to share our research journey in myopia control. Next slide. Yeah, as Professor He has said, so myopia is associated with potential long-term ocular complications that could cause permanent visual impairment, especially among the high myopes. 10% of high myopes will suffer from some kind of macular degeneration. In fact, in Hong Kong, we found 78% of teenagers who had high myopia already developed some related retinal changes. So high myopia is a health issue that impacts on the quality of life of an individual. Next slide, please. Myopia research at the School of Optometry, the Hong Kong Polytechnic University, has started more than 30 years ago. Building on our earlier prevalence studies and animal model testing, our two inventions, the disc lens and the DIMS lens, both won the Geneva Invention Award. The DIMS lens was co-developed with Hoya Corporation and awarded the Geneva Invention Champion Award in 2018. And just last month, we published the long-term 6 years results of DIMS lens and provided the evidence that DIMS lens can continuously control myopia progression and is safe to wear. Next slide, please. We use this simple illustration to show how lenses can affect eye growth. Next. By using a minus lens to shift the image behind the retina, the eye compensates the displaced image by extending the length of the eye. The minus lens induces a hyperopic defocus to the retina. Next. And by using a plus lens to shift the image in front of the retina, the eye compensates for the displaced image by shortening the length. This plus lens induces a myopic defocus to the retina. Next slide, please. Our research team hypothesized the natural process of ametropization in human eye is regulated by the equilibrium between the opposite hyperopic and myopic defocus. By providing a clear vision and at the same time project on the retina a constant myopic defocus, for all viewing distances may retard myopia progression. Next slide, please. To confirm our hypothesis in the human eye, we designed the DIS lens, a contact lens that myopic defocus was incorporated into the lens. This shows the design of the lens that there are concentric rings that alternate between the vision correction power and also the myopic defocus power, so that at the same time, both powers would be in the pupil area and provide the signal for the treatment. Next slide, please. The 2-year RCT results shows an overall retardation effect of 25%, but for children who have worn the lens for more than 8 hours per day, the retardation effect reached 60%. Next slide, please. We know contact lenses always stay on the cornea and follow the eye rotation, as shown in the left figure. But with spectacle lens, the different parts of the lens will be used for different direction of viewing, as shown in the right figure. So, next please. So if we want to apply the concept to spectacle lens, the key of the design is to make sure optical defocus must be present at all the regions. So this diagram shows that the multiple evenly distributed tiny segments of myopic defocus. And if we use the pupil location here, there are 3 illustrations. And at each of the location, we have to calculate the amount of myopic defocus and the vision correction power to be more or less equal. Next, please. So the evolution from the DIS lens to DIMS lens was through collaboration with Hoyer. Next. That the DIMS lens has finally completed its design. We shift the design from concentric rings to multiple segments. Next slide, please. And there are around 400 tiny multiple segments of +3.5 diopter in the DIMS lens. It is these multiple segments that stimulate the retardation of eye growth. And next, please. At the end of the 2-year RCT, the lens showed 52% efficacy for slowing myopia progression. And next. After the RCT, we offered to extend the trial and follow up for 1 year. And in the DIMS lens wearers group, after 3 years, these children continue to show good myopia control effect like the first 2 years. And next. And for children who had switched to DIMS lens for 1 year, myopia progression has slowed down significantly. Next, please. And despite the outbreak of COVID, we were able to recall the participants back for the 6 years follow-up. The same measurement protocol applied. And here you can see that children who wore the DIMS lens for whole time of 6 years, myopia control effect was sustained, and the overall increase in myopia was merely -0.92 diopter. Next, please. So 22% of children did not show any myopia progression once they started wearing the DIMS lens. Next. And in this figure, the 3 curves at the bottom showed the annual myopia progression among Asian children of different age in the general populations. And those curves near 0 point are the one from the DIMS lens wearers. So we can see that wearing DIMS lens significantly lower the myopia progression when compared to the age-matched population. Next, please. In summary, DIMS spectacle lens sustained the effect of slowing down myopia progression and axial elongation in children throughout the 6 years. And 22% of children did not have any myopia progression at all. 92% of children responded to the treatment with varied effect. And for those stopped the DIMS lens, treatment effect was retained and there was no rebound effect. And to our best knowledge, this is the longest follow-up study of a myopia control spectacle lens. Next, please. It is encouraging to witness that both DIS and DIMS lens retard myopia progression. The DIMS lens can continue to slow myopia progression throughout a longer period, and both lenses are now available in the market. This lens is marketed as DIS one-day disposable contact lens in Hong Kong and China, and DIMS lens, since its launch in 2018, is available as MioSmart in over 30 countries and places. And as of today, over 4 million of lenses have been sold. And thank you for your attention.
Thank you, Professor Lam. Extremely impressive. 4 million lenses. That's just amazing. So, given your results, your recent long-term results, should all myopic children be given these lenses, irrespective of whether they're progressing or not?
Yes, there have been evidence showing that before a child becomes myopic, there are already changes in the ocular anatomy that we can pick up. And there are also been reported risk factors, like whether the child's parents are myopic or not, and whether the child have too much close time, work, or not enough time outdoors. So all these factors can add up to predict a child whether he will become myopic or not. So with all these risk factors calculated in, then we can predict some children who may become myopic. So myopia management can start before they are even becoming myopic.
Very exciting. Listening to your talk and seeing you going from a basic science idea all the way through to 4 million lenses being sold, clearly that research journey has had some challenges. Would you care to share with us some of the difficulties that you faced along the way?
Yeah, we have ups and downs during the journey. And I think the most difficult part is maybe just personal for me is that it's the lack of technology to support the ideas. So when we designed the DIS lens, and there were no laboratories in Hong Kong that can help to make the lens. But fortunately, when we received the research grant, and right at that time, it's so timely that there was a Hong Kong laboratories that had bought the most expensive machine that can make our lenses and offer us a very good rate. And the same applied with the DIMS lens. So when we had our first meeting with Hoya did not promise that they can make it. They found it difficult, but they did not drop the idea. And after 6 months, they came back and they said they can do it. So that's how we started the DIMS lens clinical trial and finally launched the lens.
Very interesting. So sitting right at the intersection between vision science and new technologies. So just one very final question. Where are you going next with this line of research?
Yes, so from the clinical trials, we have new findings that can differentiate children to be good and bad responders. So we will be working on newer design to cater the different characteristics of ocular features among these children and hope to provide optimal myopia control for them. And of course, we will also test the lens on the pre-myopic children to evaluate if the lens can prevent myopia.
Fantastic. Thank you very much, Professor Lam.
Thank you.
Thank you very much. I'd now like to pass to Professor Jones, who will share with us on contact lens-based ocular drug delivery, opportunities and challenges. Professor Jones, over to you.
Thanks very much, Ben. Yes, and now for something completely different. No myopia in my talk at all, actually. If we go to our next slide. So when a patient has any form of ocular disease, the way that we as clinicians treat them in the vast majority of cases is to use topical drops. We basically take a drop with an active agent in it, and we prescribe that for the patient, give it to the patient, they go away and use it. The problem is that actually treating ocular disease using drops is not a very effective way for a number of reasons. First of all, patients are notoriously poor at actually being able to get the drops into their eye. Many people, when they try and get the drop in their eye, end up missing. If they don't miss, many people actually instill too much drop. So they end up squeezing the bottle too hard, too much drop gets into the eye, there's lots of overspill. In those situations where patients are using drops, the other major problem is that often they're not very good at remembering to put them in. We know from epidemiological studies that even in situations where patients have an ocular disease where they know if they don't put the drop in, they're actually going to lose vision, for example, in glaucoma, only about 50% of patients are actually instill their drops. In those situations where the drop actually does get into the eye, so the patients remembered to do it and they actually get the drop in the eye, the next problem is that that drug is actually diluted very quickly. We have lots of tears on the eye that dilute it, and we have lots of blinking, which washes the drug away into the drainage passages that sit in the inner corner of your eye. As a result, the drug actually doesn't stay on your eye for any length of time. Now, if we can just have a click, please. What we know is that we could enhance the length of time that the drug actually stays on the eye by increasing its viscosity. And in this report here, what we see is 3 different drops of 3 different viscosities. The problem is if the drop is too viscous, the patient ends up having vision problems when they put it in. And even in the situation where we have a very viscous drop, which is the green dots here, we can see that it only stays on your eye for around about 20 minutes at most. The next problem is that even when the drug does stay on your eye, the blood vessels that overlie the eye in the conjunctiva, those conjunctival blood vessels, actually absorb much of the active drug and move it into the systemic system. So overall, it's estimated that only about 5% of the drug that you put in your eye actually acts on the target complication. So not very efficient. Next, please. Now, what about using a contact lens as a delivery vehicle? What about if we were to take a contact lens, actually load it up with the drug and put that onto the eye? Next. What we know is that that soaked contact lens will release a drug much more slowly than a drop that's put onto the ocular surface for actually many hours. So unlike minutes with drops, we can extend that for many hours. Next. That concept is not new. It was actually suggested by the inventor of soft contact lenses, Otto Victorly, in 1961. In his original patent for a soft lens, he said that a soft contact lens could actually be used as a drug delivery vehicle back in 1961. When you survey clinicians about whether they feel that actually using a drug delivering contact lens would be useful to them, more than 90% say, yeah, that would actually be quite useful. Next. And that clinical success actually depends upon 2 things. Can we load enough drug into the lens? And we have good evidence about the fact that you actually can. The major challenge actually relates to the release of the drug. How can we slow the release of that drug onto the ocular surface? Next. Because when we actually study that, when we actually study the interaction between lenses and drugs, what we find is that the major problem is that that drug gets released far too quickly. It actually gets released. There's this bolus of drug that gets released, and extending the delivery time over a long period of time is the major challenge. Next. So in terms of slowing drugs delivery to the eye, there's basically 5 different concepts. The first of which is the one that my research group has looked at most of the time, and that's called molecular imprinting. And that's where you actually, when you're casting the contact lens, actually make cavities within that contact lens that molecularly are the same as the drug that you're trying to release. The next thing you can do is you can incorporate that drug into a film and then cast the contact lens around that film. Most of the time it's a PLGA film. Another one uses what we call diffusion barriers, and most of the work in this area is actually looked at releasing drugs more slowly through coating the lens with vitamin E. And the final concept for contact lenses that's been most well studied is a relatively simple concept where we take a negatively charged contact lens material, incorporate into it a positively charged drug, and that interaction slows the release onto the eye. Next. A more recent method has actually looked at using inkjet printing onto commercial lenses. So you take commercially available lenses and onto the surface of the lens, you inkjet print the drug. And again, that slows the release onto the ocular surface. So given those 5 concepts, do any of them actually work? And have any of them actually come to market? Next. Yes, it has, but very recently. Think about the fact that Otto Victorly in 1961 said, hey, you know what? We could actually use drug delivering contact lenses. Well, it took till 2021 for the commercial reality to happen. In 2021, Johnson & Johnson released their AccuView TheraVision with ketotifen lens onto the market. Ketotifen is a topical antihistamine that's used to relieve the itchy eyes that you get with allergic conjunctivitis. The lens uses a daily disposable platform. So each day, the patient puts onto their eye a brand new contact lens that's incorporated with ketotifen. It's a hydrogel lens. The drug is preservative-free, and that's very useful because many patients with allergies actually have preservative sensitivity. The product is released over a 5-hour time frame. So remember, that's 5 hours that the drug is in and around on the ocular surface, compared with minutes, as we showed earlier on, with topical drops, and it provides relief for up to 12 hours. So finally, we've seen that commercialization of this concept, and there's a lot more concepts of these coming along now that we've seen a regulatory pathway from Johnson & Johnson. Next. In terms of some of the critical challenges, next, now that we have seen this come to market, next. The regulatory approvals and studies required to obtain licensure can be quite complex. It's difficult to know whether these products will be treated as drugs or devices, and most of them appear to be treated as what we call combination devices, but in some instances, it's quite a difficult pathway through this combination device. Next. Some of the other considerations relate to production challenges. How easy will it be for contact lens companies producing contact lenses to incorporate into their existing production systems this incorporation of drugs? And if they need to spend multi-millions of dollars, that's probably going to be a challenge. There are the slow leakage of medications over the time that the lens is stored. So releasing from the product into the surrounding blister pack, that may limit their relevant shelf life. And finally, it's who'd be licensed to prescribe these lenses, because in many jurisdictions, contact lens practitioners who can prescribe contact lenses are not licensed to prescribe therapeutic drugs. So with that, I'll say thanks very much for listening, and I look forward to having a chat with Ben about some of the challenges.
Thanks very much, Lyndon. Fantastic presentation. So I guess the first question that springs to mind is what eye conditions are going to be most amenable to this sort of drug delivery technology? You mentioned allergic responses, but beyond that?
Yeah, and you can kind of think about these really in terms of two factors. I tend to think about this treatment as being, is it a treatment for a short-term problem, or is it a treatment for an ongoing problem? So when you think about short-term problems, things like scratching your eye. I don't know if you've ever scratched your eye, but it's absolutely painful as heck. And so having, very often what we do now is we actually treat that by using what we call a bandage contact lens that we put in, and often the patient correspondingly at that time needs an antibiotic or a steroid to dampen down the inflammation. So that will be short-term management. Things like microbial keratitis, where you get a severe eye infection, you need to load the eye up with an antibiotic. That, again, is only treatment for a couple of weeks at most. So there are those short-term managements, and then there's the ongoing longer-term managements. So patients with recurrent uveitis, patients with allergic conjunctivitis, but probably one of the biggest challenges is glaucoma. Patients develop glaucoma typically in their late 40s, early 50s, and then they have it for the rest of their life. So the treatment for those two types of conditions, quite different in terms of the type of drug you need and how long you need to release that drug to the eye.
Interesting. And I guess the other point that came to me as you were talking, the regulatory pathway is obviously complicated. Are these technologies contact lenses, or are they drugs from a regulatory perspective? Are they devices or drugs?
Yeah, and that's been a challenge, because each regulatory authority is tending to think differently. And what's tending to happen now is that, as I mentioned in the talk, people are going down this combination device. So it's a device that delivers a drug. And so typically that there's not an easy pathway to market, because for many regulatory agencies that actually doesn't exist. It's a new challenge. And so that's certainly one of the challenges. The other challenge relates to actually conducting the study. Think about that microbial keratitis that I talked about. We know that treating it with heavy antibiotics over a long period of time, it's a pain in the neck to begin with, because you've got to really load up the eye with an antibiotic to begin with, and that's a challenge. And then you've got to, put that drug in many times a day for several weeks. How do you conduct a clinical trial where you go to a patient, well, we've got this known treatment, it's kind of a pain, but we know it works, versus we've got this experimental treatment, and if it doesn't work, you might actually lose vision. it's going to be very difficult to get ethical approval to actually conduct that study and then to enroll patients as well when there is such a significant risk. So it is a challenge bringing some of these things to market.
Interesting. Thank you very much, Professor Jones. That's excellent. And I think now I'd like to open up for a broader panel discussion around some broad issues in this space. And I guess the first question relates to the importance of this whole topic of eye and vision health. And I know that all three of you, as well as being extremely successful researchers, you also see patients. And I wonder if you could comment on why we should care about eye and vision health. What happens when we lose or we experience vision impairment? Maybe Professor Lam, you'd like to kick us off?
Yeah, I think ignorance is the sin. So when we see patients, we came across many patients' parents that they do not really care of this condition, the eye condition. They think that they will handle themselves naturally. So sometimes time is wasted. So I think prompt and timely treatment is very important, and it is our responsibility to deliver this. So public education, our continuous research and advancement in eye care is very important.
Thank you. Professor Jones?
Yeah, I was fascinated by the presentations on myopia control. I mean, what great evidence about the fact that now we have really good ways of slowing the progression of myopia. I think part of the challenge for trying to get this adopted broadly is that we are trying to provide evidence about the fact that we need to slow your child's progression now to stop disease that they may not get for maybe 30 or 40 years. And that's quite challenging. We've never done that before. We've never had those kind of conversations with patients where we've said, your child is going to develop high levels of myopia. And that's not just an inconvenience because they're going to need to wear thick spectacles. Yeah, that's a problem. But, we've got pretty good high index lenses these days. It's because we're trying to stop disease in later years. And that's hard, I think, for people to contemplate. So I think that's one of the big challenges. I think we now, without doubt, have good evidence about the fact we have great ways of slowing the progression of myopia. And I think practitioners are on board with the fact that they need to get involved in this. I think myopia control is going to become the standard of care within clinical practice. In Canada, we've already had that stated by the Canadian Association of Optometrists. Myopia control is the standard of care. You have to have this conversation with your patients. The problem is parents then adopting it because it's like, yeah, but that's a problem that might not happen for decades. So I think that's a big challenge.
Yes.
Thank you. Professor Hell?
Most of the time, I just, when I practice in Melbourne, we like the very simple patient like cataract surgery, because after you've done surgery, patient come back for the first very short period visit, follow-up visit, review, and then they disappear. But then when we try to manage this myopia control, is completely different story. You have to have a very strong commitment, engagement, right? Let me say like strong engagement with the family. You have to convince the parents. You have to put the candy in your pocket, and then you can communicate with the child as well. So it's it's a different way to practice. But then but another challenge is in different part of the world, like for example in Melbourne, Australia, our optometrist is allowed to prescribe. But in some part of the world, we're not allowed to prescribe. Or then that's why we create a lot of like this one-stop management for myopia is becoming a bit difficult. For example, if we separate optical control and eye jobs, I mean, then it's quite challenging. So, but in general, I think myopia control is a long-term business. You really have to be a good communicator. You're ready to be communicate with the parents, engage them, and then it's like a couple of years effort.
Extremely interesting. Thank you. I mean, from my research area, one thing that's really struck me is how even a small change in someone's vision can have such a profound effect on their life. If it happens early in childhood, it affects their eye-hand coordination development, their educational achievement, later in life, mobility, social interaction. So the types of technology you're describing are critical for providing quality of life throughout the lifespan, I think, in this area. Thinking about your research programs, obviously, we all want to keep our research and development and commercialization programs running. How do you think we can attract and nurture new talent in the research and development field? Maybe Professor He, I could start with you this time.
Yeah, great question. So whenever we try to attract or bring in talented postdoc fellow, and then I think we got to send a very clear message to them, to the potential candidate, that you're coming in, we expect you not just running research program as like a traditional way, like a bench or deal with the cell and animal, but we want something come out of it. It's a commercialization. It's not necessarily to commercialization, but it has to be translational. It has to be a new solution for the eye diseases. Ideally, if it can be as successful as Carly and Professor Lam, that would be fantastic, selling 4 million products. But then we shouldn't expect every single postdoc fellow achieve same level of success. But as long as they understand entrepreneurship is important, and then I think that is a correct message we should send them.
Thank you. Professor James?
Yeah, I think one of our jobs, honestly, in academia these days is to bring in people who have an understanding of the fact that it's changed. So academia is not just these days about sitting in a library thinking about things and writing esoteric papers that are irrelevant. That's not what today's academia is about. We have to, as Professor Hayes just said, we have to provide education for these trainees to understand the fact that what we do in the lab has to be translatable to the outside world. So it's got to be relevant. It has to have, in most cases, at least some opportunities to do good for the world. So to be able to move that from the bench into the laboratory and then out into the real world is really important. And also it's important because there simply aren't enough academic opportunities for the trainees that we have these days. They're not going to go into academia. We're producing too many PhD students for the numbers of professorships that are available. So very many of them will end up working in the world outside of academia. And in order to do that, they need to understand the importance of that translation and bringing things to market.
Thank you. Professor Lam?
I think I always start with a research question for my postdoc and PhD students. So I think it is important to focus on a problem that affects the world and that would tackle it and be able to bring benefits to the world. So I think the research question and the passion in that research area is very important. But I also agree that nowadays to become an academic has also the talent or the skill set of translational into a commercial product or a protocol of some kind. So I'm very pleased that with the setup of CEVR, we now can kind of ride on the facilities from the science park. So they offer a lot of different programs for young researchers to learn about how to set up companies, how to get angels fund, etc. So I think this is something important now for future researchers, that they need to be equipped with these skill sets.
Thank you. Yes, and I completely. Yes, thanks, Carly. Yeah, I completely agree, and I think the InnoHK initiative has really provided us with resources to take the research out into the real world, partner with commercial organisations to have an impact both in Hong Kong and beyond. And I'm enjoying working with all of you, and I'm looking forward to continuing to work with you as you develop all of these extremely exciting and impactful technologies. So thanks very much to the panel for part one, which I will now draw to a close. Thank you again. Now we will move to part two of our thematic session, which is on the topic of retinal degenerative disease from early detection to treatment. For our panelists, I'm very happy to introduce from the Institute for Quantum Computing at the University of Waterloo, Dr. Dusan Saranak, and from the School of Optometry at the Hong Kong Polytechnic University, Professor Henry Chan. I would now like to invite Dr. Saranak to share with us on the early detection of age-related macular degeneration by quantum imaging technologies.
Okay, Hello, everyone. My name is Dusan Sarnak, and I'm a senior technical lead of transformative quantum technologies at the University of Waterloo. And today I will be talking about a recent development in integrating some quantum tech into vision science. Our collaboration now spans three major institutes, namely the UW Institute for Quantum Computing, the UW School of Optometry and Vision Science, and Center for Eye and Vision Research in Hong Kong. Next slide, please. The first target technology that we focused on is related to structured waves. There's an entire toolbox that has been developed for creating special forms of custom light fields. The backbone idea is that you pattern the wavefront, and if you do it cleverly enough, you can get some unique properties to emerge. On the leftmost side are examples of phase and polarization patterning that are the most common. One important case is the Airy beam that shows a curved trajectory in free space, and it is shown in the middle. So whereas the light beam typically travels in a straight line, if you induce a cubic phase, you get this beam whose intensity follows a parabolic trajectory. Furthermore, it possesses a self-healing property, as shown if you place an obstacle in the path, the area beam will actually heal after the obstacle, even though it was obstructed. As you can see in the figure, this is for an electron beam, but the methods are quite general and can be applied to light, neutrons, atoms, molecules, and anything that travels as a wave. The other very important example is the case of orbital angular momentum beams, which possess helical wavefronts and carry a well-defined orbital angular momentum. As shown on the right, these beams possess a corkscrew-like shape where the light beam can be thought of as essentially spinning. Next slide, please. Structured light is relatively a new field with roughly 30-year history, but as you can imagine, it brought forth quite an array of impactful applications, given that so many devices and experiments are based on optics. Structured light essentially provides a new degree of freedom, and new types of probes can be tailored for more specific and more selective. They can be more specific and more selective towards particular applications. The main applications so far include high-resolution optical imaging, high-bandwidth communication, quantum information science, and optical metrology. In 2020, our group made the first integration of structured light in vision science, and in particular, our work was taking advantage of the polarization topologies that are manifested by helical beams with quantized orbital angular momentum. Next slide, please. The application we looked at relies on the human ability to perceive polarization of blue light. And this ability is enabled by a series of radially symmetric dichroic elements, macular pigment, that is centered on the foveola in the human eye. Their orientation effectively forms a weak radial polarizer within the eye, and therefore when a person with a healthy macula observes uniformly polarized light, they can perceive a faint entoptic phenomenon consisting of two azimuthal fringes that is known as the Heidinger brush. And if anyone is ever interested, they can observe the Heidinger brush either on a phone, or if they put up a blue screen, or if they look at the clear sky 90 degrees away from the sun, you will notice that there's a Heidinger brush, a very faint one that always points to the sun. This phenomenon has been exploited in studies of central visual field dysfunction and age-related macular degeneration, characterizing macular pigment density and determining the location of the fovea. The most important example is the early detection of AMD, as it is a global leading cause of blindness among people over the age of 60. The loss of ability to see the Heidinger brush has been associated with the early signs of AMD, but one of the main hurdles is that this is the very relatively weak signal of an entoptic image. It is really difficult to see the Heidinger brush, and it is quite faint. Next slide, please. The result of our work was to create much more powerful entoptic signals. By using the helical forms of light, we are not limited to two azimuthal brushes. We can arbitrarily fix the density of the entoptic lines. What is shown on the figure is the observer, in this case me, looking at a special form of helical beams that induce any number of azimuthal fringes, as shown in the bottom right. Next slide, please. This is my last slide that emphasizes the power of the techniques that we have developed. We can apply analogous methods that we develop for quantum information experiments with structured light and obtain radial fringes if we want to, not just azimuthal fringes. We can decouple the entoptic signal from corneal bifringens. We can decouple the signal from a person's eye prescription. These are all tools that have been developed for quantum processing that we essentially apply the analog to vision science applications. And we can also image the structured light that is directed onto the macula, and as shown in that example on the left, and we can directly see in real dimensions which part of the retina is being illuminated and at what strength. So we have 3 of these setups at the moment at the 3 institutes, and we look to make further progress in the near future by exploring the power of structured light in vision sciences. Okay, next slide, which concludes my talk. Thank you very much for listening.
Thank you very much, Dr. Saranak. I think I'm right in saying that this is one of the very first applications of structured light technology to vision science. So congratulations on your technology. I wonder, obviously there's huge potential in this field. I wonder if you could comment on some of the other potential applications for structured light technology and quantum technologies in general in vision science and eye health care.
So an interesting one can be the self-healing beams that I described and applying them for cataract investigations. They seem very well suited for that because cataract is essentially putting obstructions in a person's field of vision. And it would be interesting to explore how the self-healing of light would interplay with that and what we can do to explore their connection, so to say, if one can compensate for the other. Another example is quantum ghost imaging. So that technique is essentially using one wavelength for interaction with an object and another wavelength for imaging. So for an example, you can imagine a situation where you shoot infrared into the eye to interact with the retina while you use visible light to image what the infrared's interaction. So you're using one wavelength that doesn't induce a response in the eye, and then you're imaging the wavelength that you have the best detectors for.
Very interesting. And so this project you described, what are your near-term goals for the project, and whereabouts is the project positioned at the moment?
So a near-term goal is to develop a device that is sensitive enough for detecting early stages of AMD, and that right now we're testing participants with very early signs in one of their eyes and looking at how their good eye compares with their bad eye. And it seems to be rather clear that although their normal vision is not impaired at all, they function normally, they cannot perceive the entoptic profiles that we create with structured light the way a healthy person can. It's very different what they see. So the versatility of the stimuli that we can create allows us to perform a detailed characterization, and we hope that it is sensitive enough for a meaningful detection of AMD in a commercial sense.
Very interesting. So you are moving towards commercialization of this technology?
Yes. Yes.
Excellent. And just very finally, you're talking about a relatively new form of light that you're using here. Are there difficulties and challenges that people face when perceiving this light? And how is it different from the normal types of light that we think of?
So one difference is that the image appears sharp regardless of a person's eye prescription. That is because the incoming light is collimated. So the lens in the eye doesn't need to focus two different light rays onto the retina. It's always a focused image. A disadvantage, on the other hand, is that coherent light manifests something called a speckle pattern, which is essentially a spatial interference between two very close points. You can imagine it like the old TV grainy signal noise pattern that's always in the background. It's very weak, but it's always present. Safety-wise, which is what usually people wonder about, there's no difference between normal light and coherent light. The wavelength and the intensity of light determine the safety levels regardless of anything else. And there are very clear guidelines for maximum allowed exposure values when illuminating the retina with light.
Thank you, Dr. Saranak, for your presentation and for our discussion. And I'd now like to move to Professor Chan, who will share with us on neuroprotection in retinal degenerative disease, and in particular retinitis pigmentosa by Lycium barbarium. Professor Chan, over to you.
Thank you. Today, I would like to share my work related to using the Lycium barbarium in the neuroprotection against one of the retinal degenerative disease, which is called retinitis pigmentosa. And this is my study. I also have the collaborators with Hong Kong U team, Professor K.F. So and also Raymond Chang, and also with the Jimmy Lai from the ophthalmology department. Next slide, please. Let's talk about what is retinitis pigmentosa. This disease actually is a neuro a kind of gene mutation retinal degenerative disease. So the people will have the problem in the beginning with the ROC degeneration. ROC degeneration means that they will have the night blindness in their early life. So later, the degeneration will spread to cone. Cone is for the day vision. So you can see from the right-hand side, the picture, you can see the retinitis pigmentosa RP people, the retina will have some changes. You can see the pigments around the peripheral part. So it will cause the vision to be reduced in the periphery. So you can see that the people will have the tunnel vision. That means just the central can left. So if it further progressed, this tunnel vision part will also become smaller and smaller and finally become total blindness. Next slide, please. So Lucerne barbarum, actually is a traditional Chinese herbs which was been used for so many years in our community. All right. So why is this useful? Because from the Chinese medicine book, it say that it can help to improve the vision. So this is why we try to use it for this disease. And Lucerne barbarum has the polysaccharide. Polysaccharide, which contains the very strong antioxidative effect. So actually from different animal studies from our team and also from Hong Kong U teams, we found that it can help to prevent the neurodegeneration. So this is why I try to use it in the RP patients to see how it can help. Next slide, please. So the objective of this study is to try to see how we use this Lucerne barbarum supplement for a year and to see how it can help to improve the retinal functions and structure for the RP patient. Next slide, please. So the method is a clinical trial. It's a double-masked random study. So we will have the subjects, okay, and allocate into two different groups. One group with the Lucerne barbarum treatment, and another is the placebo. And all of them will have the baseline eye examination and also the follow-up every 6 months for a year. So we will do a lot of examinations to investigate the changes after they have this Lucerne barbarum supplement. Next slide, please. So in this study, we in the beginning we have around 160 RP subjects join in this study, but after the checking and also to see whether they are eligible to join. So finally, there are 50 will join this. There are 50 will join this study, and we allocate 25 subjects in the LB group and another 25 in the placebo group. And after 1 year, just 42 subjects can complete the whole study. And in the treatment group, there were 23, and the placebo group will be 19. Next slide, please. So this is the baseline data. According to, for example, the age, the vision, or other examination results, both groups, the LB groups and also the placebo groups, they're similar. That means there's no difference. And one thing I would like to mention right here is the side effect, because after 1 year, we also check whether they have any side effect within this year. And for the LB group, just one case report to have a mild nose bleeding. But for the placebo group, just report about 30. All right. So next slide, please. Now I would like to talk about the result. I just choose those results with the significant changes and let you have a look. And the first one is the visual acuity. We investigate the visual acuity in terms of high contrast or low contrast. So you can see the pictures in the bottom. So one is the high contrast chart, and another is low contrast. Okay, let's look at the result. So you can see that the blue line are the lines related to the placebo group. So you can see that the placebo group, they have the deterioration of visions in 1 year. But the orange line is the LB group. You can see that the vision can maintain in this 1 year. So it means that the LB can save their vision. So next slide, please. So this is the related structure. We use the OCT, optical coherent tomography, to measure the fovea thickness because we would like to look into the retinal structure changes, etc. So you can see that the same. The blue line is the placebo group. So you can see in 1 year, the fovea thickness becomes thinning. But the LB group, the orange line, you can see they maintain the thickness. It means that the retinal degeneration seems to be minimized under the LB supplement. Next slide, please. So this is the comparison between different types of clinical trials. Actually, the first type of such trial for the RP patients started in 1993. They used the vitamin A. And later on, within this maybe 30 years, you can see that they use different types of supplement, for example, lutein, omega-3, DHA, etc. So, but if you go into the results, the visual acuity and retinal structure, we can see that the use of the lucent barbarum in our study seems to be quite promising because both visual acuity and retinal structure show the delay or minimize the changes in the treatment group. Next slide, please. So from this study, we can see that is the supplement of the lucent barbarum can really preserve the visual acuity. That's mean to make the RP patient to delay the vision deterioration, and also preserve the retinal structure. That's mean the fovea thickness, everything seems to be maintained. And because of this, we believe the lucent barbarum can be a potential supplement to help to delay the visual deterioration in this group of patients. Next slide, please. So right here, I would like to thank you the support from the Health and Medical Research Fund, and also Retinal Hong Kong for the subject recruitment, and also Yuanshan Limited in Hong is a local manufacturer to supply the lucent barbarum, and also my research team. Next slide, please. Thank you very much.
Thank you very much, Professor Chan. Extremely exciting results there from your trial. I wonder if you could comment again. You did mention it in the presentation, but why did you choose this particular herb for the study?
Actually, interestingly, because in the beginning, I talked to Professor Kev So-called Fai from Hong Kong. He is a very famous scientist in Hong Kong, worked in neuroprotection. So he mentioned that the lucent barbarum is a good choice for us to try because in the traditional Chinese medical book, it seems that it can help because apart from vision, it can also help the liver. All right. It is also from the Chinese book because the medicine book, they say that liver also help the vision because they have link. All right. This is the Chinese medicine knowledge. All right. So actually, Professor So asked me to try, and I try on it in the beginning, not the human. We try in the animals. We found that it is quite useful. Even the axons can be regenerated. So this is why we found that it is useful. So after around 10 years, we looked into that. So finally, we try on human to see whether it can be used. All right. So this is why we tried the lucent barbarum. And also, in terms of price, this is not too expensive in the Chinese herbs. Yeah.
Very interesting. Thank you for Professor Chan. Wonderful work. I wonder if now we'll move on to some more general questions for an open discussion.
Yeah.
I'd like to ask you both on your thoughts for opportunities in innovation, technology, and investment in the eye and vision health field. Perhaps Professor Chan, if you'd like to comment first.
I think you mean opportunities in the technology. I think it is very important to have more opportunities in Hong Kong, especially in Hong Kong, because Hong Kong close to China. Now you know Great Bay Area has a very fast and strong changes because the technology and also the development is very fast. So I think in Hong Kong we need to use this kind of opportunities to help us to improve the eye care, because I think that in terms of eye care in Hong Kong, the development is good, but can be much faster and can be go into a better way. This is my thinking, and especially, for example, just Dr. Dusen, what he worked on the structure, the coherence, light, etc. Because I think that this type of technology, we can use it and then to apply in not just the ARM, I think in other areas, because I just think that maybe it can help to be a therapeutic light. I think light therapy, because this is also I mentioned with other professors in Hong Kong, because light can stimulate the eye. So whether it can also to make the eye better. So this is my thinking. So I think we can apply this type of technology. Yeah.
Thank you very much. Dr. Saranak, any comments from you? I know that you're coming from a very different field into this research in eye and vision health. What do you think of the opportunities for new technologies and also for investment in the area?
Yeah, so to add on to the previous answer, so as you say, I'm a physicist and an engineer more than somebody with a vision science background. So speaking from my experience with SEAVR, for example, I was fortunate to even visit SEAVR recently as the travel restrictions have been lifted. And it was really, for me, it was quite wonderful to see what has been put in Science Park, and which looks like an environment that encourages and pushes forward innovative discoveries. I think it's exemplary. And overall, the collaboration has really given me firsthand experience about the impact of the research on a global scale and the global importance of advancing technologies that can be used for diagnosing retinal diseases, for example, such as age-related macular degeneration. Likewise, the exposure to the available investments, which is also new to me, that has been put forth, it's quite substantial, and I think it will drive advancement. Basically, it will drive the advancement to bring our research technologies from the lab into commercial applications and out there to the public.
Thank you, Dr. Saranak. I agree. I mean, I think that obviously there's a huge market in this area as well. Almost all of us will experience some kind of vision loss at some point in our life. And so these are extremely important problems for us to solve. Well, thank you to our panel. And I'd like to thank all of our speakers today for their outstanding presentations and discussion. The technologies that have been described highlight the importance of collaborative interdisciplinary research for advancing eye and vision health, and are examples of the 25 projects that are currently underway within the Centre for Eye and Vision Research. As an InnoHK Centre, we welcome collaboration with academic, health and commercial organisations as we advance our mission to generate technologies that prevent vision loss and preserve healthy vision. And thanks to all of you very much for attending today.